So thank you very much for the opportunity to join you again. Today, I'll be speaking about the impact of new variants on global epidemiology, disease acquisition and progression as we continue to monitor the SARS-CoV-2 pandemic. So I thought I'd start, and I've given a few of these talks over the years, and I seem to always start the same way, but I thought I'd start with a slide taken from Our World in Data, which is a great resource and enables you to visualize very clearly where we are in terms of SARS-CoV-2 variants of concern and which ones are dominant at which stage. I'm starting here with data from the United States, as you can see, and what this graph shows is that the proportion of each of a series of variants, with each color here indicating one variant. And you can see shifts in the colors over time from May 11th, 2020 through to November 20th, 2023, with each one of those colors representing a new emerging variant of concern. So if you look towards the end of that graph, you can see that if you look at all sequences that come out of the United States recently, about 50% of them are that kind of dusky blue color, which denotes a new variant of concern, newish variant of concern, called Omicron EG5.1, which is going to be one of the major focuses of the talk that I'm going to be giving today. So this is where we are in the US, approximately 50%, as I said, of new infections being caused by EG5.1, but it's not entirely dominant. As you can see, there are still other variants co-circulating in the US. And that's very much true of the rest of the world. I'm here showing you sequences from two other countries, as far apart from one another as I could get from the US. Here's SARS-CoV-2 variants in analyzed sequences from China up at the top and from Australia down at the bottom. And you can see just eyeballing these slides that they're pretty different patterns. In general, most of the world has seen most of the same variants, but the timing with which a variant sweeps across the country can be quite different. If you just look, for example, at the light blue here, you can see changes in timing of that variant between China and Australia. And the same is true when we look at Omicron EG5.1, which is the variant that I'm very focused on in today's talk. You can see that in China, for example, EG5.1 dominates the epidemic currently and is responsible for about 90% of new infections. In contrast, in Australia, down at the bottom, you can see that it's rapidly emerging. You can see there at the bottom a big burst in that dusky blue color with Omicron EG5.1 accounting for about 50% of sequences, but it's emerging in the context of many other variants that are waning at the moment. And so slightly different kinetics and slightly different variants, but very much the same overall picture if we look globally. This is another way of looking at the same sort of data, here looking at SARS-CoV-2 sequences by variant, in this case putting some numbers on the number of sequences that are used to define these lineages. But you can see here data for Australia, Germany, Canada, Italy, Belgium, the United Kingdom, the US, France, and Spain. And you can see two things. The number of sequences is very variable, and I'll come back to that point in a couple more slides. But the other thing is that, again, the pattern is slightly different depending on where you look, representative of the differential spread of variants, different rates at which the variants are spreading in different population immunity, and also a lag phase. You know, some countries see rapid expansion of a variant before others. But at the moment, in all of these countries, the globally dominant variant is EG5.1. South Africa, where I'm based, is slightly different in terms of the profile of emerging sequences here. These are two pie charts examining the clade specification for September and October, based on somewhere between 100 and 200 sequences per month. In these slides, EG5.1 is shown in the purple color, and you can see that in South Africa, it's not the dominant variant of concern currently circulating. In South Africa, it's another variant, also a focus of this talk, called Omicron BA.2.86. And another very interesting variant of concern that I'm going to spend quite a lot of time on in this talk. But I wanted to make one quick point before I go on to speaking about the characterization of these two interesting variants, which is that our understanding of what is currently circulating in the world is very much determined by how much we're testing and how much people are testing when they feel ill, and how many of those tests then go on to become sequences which are deposited into Gizzade and analyzed by the world. I showed you that BA.2.86 is pretty dominant in South Africa, that orangey brown color down at the bottom. But if you look at the graph at the top, which doesn't show you proportional percentage like the one at the bottom, it shows you absolute numbers of sequences. You can see that the numbers of sequences upon which we base our analysis of variants in South Africa is actually very low. For example, you look at the yellow peak here, you can see we had a huge number of infections and a huge number of sequences. And so we were confident in saying that most of the circulating variants there were Omicron BA4. But our clade assignment currently in South Africa, and I'm using this as an example of many other parts of the world, is based on relatively low levels of sequencing. And this is becoming increasingly the real world. People are not so very much interested in sequencing anymore, and fewer people in many parts of the world testing. So the role of genomic surveillance is very much dependent on that testing, and our knowledge is only as good as the number of sequences that we have. So I said I was going to spend much of the talk speaking about these two variants, and what I've done here is to summarize the state of the art, essentially, the most recent papers describing the features of both BA.2.86 and EG5.1. This figure is taken from a publication by Riel-Losanier, and what it shows is the mutational profile of EG5.1 down at the bottom. It's compared to BA2 in orange, BA5 in the darker blue, XBB1.5 in green, and then down at the bottom you can see the new variant EG5.1. And you can see that it's very similar, in fact, to XBB1.5 in terms of its mutational profile. So here, each of the boxes is a mutation. And if you look upwards, you can see the mutations are located either within the N-terminal domain, the RBD, SD1 or 2, or the S2 region. If you compare XBB1.5 and EG5.1, you can see that many of the mutations are very similar, and that reflects its evolutionary history. But there are two significant mutations that distinguish the two variants. You can see them there at position 456, and I'll show you a slide in a second that makes it easier to see, and at position 52. This is a very similar schematic from Dan Baruch's group, which also shows the mutations and shows the evolutionary progression that resulted in EG5.1. So here again at the top, you've got the N-terminal domain and the receptor binding domain, and then you have the mutations that define BA2, which, if you recall, we considered hugely mutated when it emerged. And that's compared to XBB1.5, EG5, and then EG5.1. And you can see more and more mutations that define these different lineages. Down at the bottom in purple are those two mutations I spoke about that define EG5.1 and that are significant, at least they look significant, and I'll show you data that confirms that in terms of mutational escape. So that's EG5.1. The other subvariant, the one that I mentioned particularly emerging in the context of South Africa, but now spotted across the world, is a variant called BA.2.86. This is a highly mutated subvariant. It was identified in late July 2023, and initial estimates of its effective reproduction number, which is a measure of how fast we might expect it to spread, suggested that this subvariant was 1.3 times faster than XBB1.5 and near equivalent of EG5.1, which is, as you saw from the data I showed in the first couple of slides, rapidly expanding. And since the emergence of 286, multiple countries have reported its presence, including an outbreak that received a lot of attention in a British care home with an attack rate of more than 85%, despite the fact that most of the residents of that care home who were infected despite having recently received a beta variant-based subunit vaccine. So initial indications that it might be problematic, and that's why it was flagged very much to the community. This is a phylogenetic tree, and I'll take you through it for those who don't normally look at these trees. But what it shows is that BA.2.86 likely evolved from BA2, which is kind of interesting because BA2 hasn't circulated for a very long time, actually. The geographic location of where BA.2.86 likely popped out from is not currently known and may never be known. But as of September 20th of this year, BA2.86 sequences had been detected in at least 20 countries, ranging from Australia and Belgium and Canada all the way through to Thailand, the UK, and the US, as you can see from that long list. But what is kind of intriguing about 286 is the long branch length. So what I'm referring to is the fairly long distance between the node of the tree, which is BA2 on the far left-hand side, that purple dot. And then you can see there's a long line moving from left to right before the red arrow, which denotes BA2.86. And what that means is that the many mutations that distinguish BA2.86 from BA2 were not observed in the sequencing data. It was this big period of unobserved evolution, which is very similar to what happened right at the beginning of the Omicron waves, because there have now been many of them. Omicron essentially was detected having picked up those 30 initial mutations that defined Omicron BA1, but the intermediates had not been observed in the sequencing data. And the same is true here for BA2.86. We know it likely evolved from BA2, but the mutations that gradually were accumulated by BA2.86 were not observed. And that again speaks to the lack of general sequencing globally now, is that those intermediates likely occurred and likely spread within the population, but we didn't see them in our genomic surveillance. I also wanted to just point out, and this is a statement from this paper by Daniel Schurd at the Karolinska Institute that points out this kind of striking thing that BA2.86 is now one of more than 1,500 known variants of Omicron. It really speaks to the amount of evolution that's still continuing just within Omicron. BA286 is shown down there at the bottom, has 60 amino acid changes relative to the wild type. And you can see that many of them, the ones highlighted in dark blue here, are unique to this variant, distinct to BA2.86. So it's really highly mutated, even compared to EG5.1, which I've already spoken about. And that really did alarm many people. And so a lot of work was done quite quickly to understand this variant. This is, again, a similar schematic, and I'm not going to spend very much time on it, but it does identify exactly the mutations down at the bottom in red, which distinguish BA2.86 from previous variants of concern. So the question is, for both EG5.1 and for BA2.86, what do all these mutations mean in terms of sensitivity to antibodies? The initial hint of what they might mean comes from these kind of three-dimensional structural models, which visualize the location of mutations on the envelope spike, which is the target of our neutralizing antibody response. So most of our antibodies target spike. On the left-hand side is highlighting the immunodominant epitopes of SARS-CoV-2. And these include the RBD, the receptor binding domain, multiple regions in their class 1 to 4, as well as the N-terminal domain. On the right-hand side, you can see a series of comparisons between variants. And if you look down at the bottom at panel D, a comparison between BA2.86 and XBB1.5. And there, the blue indicates mutations in XBB1.5 that are not present in 286. And the red is new or different mutations in 286 compared to 1.5. But you can see many of them are located in those regions that are immunodominant, and that is very much indicative that they would confer escape from antibodies. And indeed, they do, both of those sets of mutations. This is, again, data from Maria's paper, and essentially what it asks in 3 different sets of serum blood taken from people who are either infected with XBB1.5 or with BA4.5, which has the same spike, or people who received a bivalent BA4.5 booster. The lower the bars here, the more resistant the variant is to antibodies. So if you look on the far left of each of these graphs, you can see D614G, the ancestral variant, nice high titers there ranging from nearly 400 to more than 1,000. But if you look at the final 2 columns in each of the graphs, that's EG5.1 and BA286, you can see a huge loss of activity of those antibodies. Those bars are much lower. And the titers are somewhere between 51 and 123. That's probably below the level of protection. And so we might expect to see lots of breakthrough infections, even in people who have antibody responses. And indeed, that is what we're seeing. But I guess also to make the point that there's not a massive difference between EG5.1 and BA2.86, despite the fact that you remember 2 slides back, there were a huge number of mutations that distinguished the 2 variants. but actually they show fairly similar neutralization profiles. This is another set of data to belabor that point. This is data from Dan Baruch's group, again looking at 6 sets of sera at multiple different time points. On the far left of each of these is the ancestral variant, the most sensitive. So if you look at the top left, that is a titer of 1 in 16,000, which means you could dilute the blood 1 in 16,000 times and still see neutralization. But if you compare BA 2.86 at the end in red, or in purple, 4 in is EG 5.1, you can see huge reductions in those numbers down to 107 and 304, which means you can only dilute those antibodies 1 in 100 and get the same amount of protection. I guess the other interesting thing is that in this set of data, and there are a few sets of data that indicate this, actually BA 2.6, despite having more mutations, is less resistant than EG 5.1. So something to factor in as we watch the spread of these variants. Of course, of interest always, when we look at these mutations, is their impact on therapeutic monoclonal antibodies. We all had huge hope for therapeutic monoclonal antibodies early in the pandemic. We have lost quite a substantial amount of that hope since the variants of concern emerged. Here you can see 4 monoclonal antibodies that are listed, and they have unpronounceable names, as do all monoclonal antibodies. And you can see that 3 of them had activity against BA2. Against XBB 1.5, only 1 of them has some activity, and against BA 2.86, no longer do we see any activity. And this is a statement out of Ria's paper that confirms that despite the huge number and very different mutations that distinguish these 2 variants, they have similar neutralization profiles to polyclonal serum antibodies, and here you can see both have very little sensitivity to monoclonal antibodies. In the last couple of slides, I want to point out that antibodies are not everything, despite the fact that I've spent my life studying them, and remind you that the part of the immune system that prevents severe disease and death is likely not antibodies so much as the T cells. This is an old Nature paper commenting on the ability of T cells to recognize the Omicron variant, which had 30 mutations compared to the 60 that we now see. Here, what Wendy Burgers and Catherine Rue and colleagues from the University of Cape Town here showed was that even against Omicron, those T cells retained activity. And so the question is, against BA 2.86, which is now twice as mutated as Omicron, do those T cells still retain activity, and can we expect that they would protect against severe infection? And indeed they do. This is unpublished data. It's on medRxiv, also from Wendy Burgers and Catherine Rue. But essentially it shows that when you compare T cells to the ancestral, this is CD4 T cells in blue, to the ancestral variant, and you go all the way along and compare them to BA 2.86, there's not a big drop-off in activity here. And panel C shows you the same thing. That's the fold loss, and you can see that there's not a lot of difference between CD4 T cell responses to BA 2.86 compared to the ancestral strain. That's CD4 cells. If we ask the same question in CD8 cells, you see a little bit more variation, a bit more movement up and down. But for the most part, even against BA 2.86 with its 60 mutations, you can see that the T cells do a pretty good job. So what we would call, and what Wendy does call, post-pandemic memory, T cell responses to SARS-CoV-2 are durable and broadly targeted and cross-reactive even to BA 2.86. And I think that's continues to be good news. And that's where I'll end. The continuing evolution of yet more variants, I think, and the low levels of testing that we're seeing highlights the need for us to continue with this genomic surveillance. We obviously missed something with 286, and we may be missing a lot of other evolution. Both of these 2 variants are highly immunoevasive for neutralizing antibodies, but not substantially more than what we've seen so far. Not much more than the XBB variants, which have infected much of the world. So that's probably good news. And post-pandemic T cell responses remain very resilient, even against 286. But I think what probably still needs to be established is the relative clinical severity of BA 286. And as I said, in the context of reduced sequencing, it's very difficult to know where we're going next with these and which of these variants will continue to dominate. And there I'll stop. I just want to acknowledge my lab and our funders who continue to enable this kind of work to happen. And I'll stop there. Thank you.